all of that just suddenly went away, because here there are other people who are just like me, and they were coping with the disease. They knew how to cope with the disease. There was knowledge in this room, and everybody there is so nice, and they help each other, and I felt like I was among friends. They're just a great group of people. They're all so supportive of each other. They're just wonderful.
I met Linda Feld a couple of years ago, and I never knew what PPA was, and I met Linda Feld, who I find one absolutely amazing individual, and she's so committed to addressing the issue of periodic paralysis and helping that next generation. I took it upon myself I told her, I said, you're an amazing human being, and if I can be any support and help for you, I'm going to do it.
Well, the PPA, the Periodic Paralysis Association, means very, very much to me because it was them and their conferences where I learned that I was not alone. I can get emotional about that because that first conference is still in my heart. It was like a family that you never knew you had, and that's where I learned about all these triggers. I never knew about all my triggers.
I have attended PPA conferences in the past. I was actually genetically diagnosed at a PPA conference when I was 21 years old. When I went to that conference, I was terrified. I was scared. I didn't want to be there. I didn't like talking about the disease. It was something that I didn't want to have. My mom forced me to go, and my brother, and it was uncomfortable. I wanted to crawl out of my skin, but here I am several years later, and I'm a patient ambassador. And so that conference changed my life in several different ways. It was a stepping stone.
Advocacy to me is one of the most powerful things that we can do in the rare disease community because of who we are. We're rare, right? We're rare and not rare at the same time. So getting more people out to tell their stories and to reach out to those who are lost is most powerful, and we can really impact people. I first learned about the PPA. The PPA has been a part of my life since I was a little kid. When my mom first learned about periodic paralysis when in 2007 or 8 or whenever she was diagnosed, and then shortly after I was diagnosed. And I remember I was like 7 or 8 years old, and we went to the periodic paralysis conference that was in Florida. It was in Orlando that year. And as an 8-year-old, I remember meeting more people and being like, wow, some of these people are in wheelchairs, but others are running around. I didn't really understand where I was, but I understood that it was an amazing community and people were doing good things. I credit a lot of what has been possible to them because they connect so many people. They were the light in the dark for my mom when she first was diagnosed.
So PPA, or the Periodic Paralysis Association, to me, that group of people, Jake Levitt is the president, they mean the world to me. If that organization didn't exist, well, I'm not sure I'd be alive, to be honest.
So, they say together we're stronger. That video was made by Strongbridge Biopharma, and they have been just such an incredible partner when they came on the scene and they acquired Keveyis. everything they've done has been on the up and up. They provide free medicine when they can. They've been doing videos like this, making their PP ambassador program. That helps people who are new to this. They connect people who are new to this with veteran folks who can guide them. They have a lot of marketing efforts that obviously, you do because you're marketing a drug, but these efforts raise awareness to physicians, they raise awareness to patients. It's not just about Keveyis, it's about periodic paralysis, and it's so important, and it's a very well-rounded, honest effort. They're just a wonderful partner, and I just want to give them a big round of applause. And just on that note, there is a dinner that they are having tonight. If I misspeak, anyone from Strongbridge, let me know. But there's a sign-up sheet outside. Everyone in this room is welcome to go. And I think they would like you to sign up just so they get an idea of who's coming, even though I know that they've planned this already. But please visit them in the back there. And then, Linda, do you want to go through who all the vendors are in the back briefly?
Oh, wow. Yeah, this is going to be hard for me to do because I will tell you that there is a group here from the University of Florida, and they are here, and they're going to be doing a survey during the conference about pain and depression, and yell to me what else it is? Huh? Quality of life.
That's a
Yes.
that's a game of telephone right there. And I think that's how we should do it. We should do table 10 to table 7 to table 2, table 2 to the mic.
I wish you all had mics back there. But anyway, they have a survey that they would like you to go back and pick up from them. They're going to be here today and tomorrow, and they will be collecting all of these by tomorrow. And they hope to do some research into those subjects, and all of you taking part is going to help them do that, and in turn, it's going to help all of us explore those different parts of periodic paralysis. And they would like caregivers as well as people who are patients to take that survey. And now, I know, let's see, we've got some people here today, and we've got some people that are coming tomorrow, and, I know there's...
The voice?
Yeah,
Rare patient voice.
Rare patient voice. They need. Do you even know what?
No. They're an outfit that kind of collects the names of and contact information of patients, and then pharmaceutical companies can then contact them and do survey. They become a third-party market research opportunity, and you can learn a lot of things, not just from doctors, but from patients. And so they're a mechanism of connecting the patients to the pharmaceutical industry, and that allows exploration of unmet needs and other things. So please sign up with them. It's I just did it. You get $10 on Amazon or Starbucks or something. Or is it Amazon? What? Starbucks, Dunkin' Donuts, or Amazon. So if you're hyper, take the Dunkin' Donuts. And anyway, so.
And then tomorrow, we will also be having an acupuncturist here. So a lot of people now are being treated with acupuncture for periodic paralysis and getting very good results from it. But it's something that you have to decide for yourself. But she will be here to talk to you about what she's able to do for people. And I know that Monica, I know, is somebody who is successfully using it. And I'm sure Monica can point out other people that are successfully using it.
No pun intended on the point.
Yeah, okay, yeah. What else?
We're going to have an auction. So we struggle every year if we should do the auction again or not. And so, but there's always one or two items that kind of warrant auctioning off. So maybe tomorrow we'll do that. We'll let you guys see the stuff, and hopefully we'll have a little fun.
And tomorrow, probably right before we break for lunch, we're going to be doing a photo of this group, which is going to be a real challenge.
That'll be interesting. It'll be interesting.
So somehow we're going to take a picture of everybody here. So if you have something you want to wear tomorrow for the picture, feel free.
Or nothing.
Or
Just-
nothing.
No, I'm
Yeah,
just
we
kidding.
can really make
No,
this
no.
picture something.
We're not in Las Vegas anymore.
Yeah.
And Shelley, do you want to just do like some housekeeping stuff? So there's where the bathrooms, if you have an attack, et cetera, et cetera. I'm sorry.
Oh, and I just wanted to say one other thing. We do have people with allergies in here. Would you please refrain from bringing any corn products or any nut products into the room? We can't police the rest of the hotel, but we just ask that you not do that for this room. Okay, Shelley, sorry.
Wow, now I can look. Did you break it? Okay, so now I can see without that light in my eyes. This is a lot of people. That is amazing. Oh, and what he said about the auction isn't exactly true. It's going to be tomorrow after dinner. We do have a couple quilts, which are amazing, but some people have brought in some things also to contribute. And it's on the back table by our wonderful EMS guys. So I don't know if you know, there are their hands. These guys are here to help us if we need anything. So one of the housekeeping issues is there are blankets and pillows along the wall. That's perfect world if we can get you there in time. If you need anything, have someone at your table, get a hold of me. I will be looking around the room trying to spot people who are going down. I'm pretty darn good at it. But if I miss someone, please let me know because we can move the pillows, the blankets. We want you to be comfortable. This is a place where you are normal, and if you don't have this disease, you are not normal here. So it's all about us, okay? It is all about us. And if someone goes down, take the opportunity to kind of check it out because it is cool. You can find out who's like you. Sometimes we have the McClonic jerking. I have flipped myself over. I think that was in Vegas. That was awesome. We're going to just learn from each other. So that leads me to the lanyards we have. They are different colors. Trying to get people as much as we can to speak with one another. You are going to learn so much from the doctors, but you are going to learn a lot from just talking to each other. What your symptoms are, what the tricks are for handling things. So the colors will kind of help guide you if you're a hypo or hyper, just anything you need. And ask. Please ask if you need anything at all from us. The bathrooms, when you go out the door, you're going to see a sign that says Conway. Conway is where the first set of bathrooms are. There's a men's and a women's right by it. And then further to the stairs, there's another set. So we should have plenty of bathrooms. The man taking the pictures, this is Uva. He is awesome. This will be, I think, your third conference. I run around the whole time anyways, but then I had my camera on top of that, and he's made my life a lot easier. So thank you. Let's see, what else do we have? Okay, so as treasurer, and I am stepping down, this has been something I've loved doing, and you'll see me at every conference, so there's really no difference from anyone in here. You won't even know I was gone. But I wanted to have a shout out to Wendy Richards. Where's Wendy? There you are. She has been signing this up through her family trust, and I just found out that she's the reason I'm getting checks, nice, lovely checks, so thank you. We appreciate that. We appreciate all donations, but that was awesome. And Kimberly and Kylene, you ladies have totally rocked the Pushing Past Paralysis. I love the effort. I love that you're giving recognition to the PPA. People are learning more about paralysis through this, and they were giving us money. I love that too. So good job, ladies. That is amazing. One of the last things I have is Amazon Smile. Who has Amazon accounts? Anybody? So Amazon Smile is just their way of giving back. You pick your charity, and you can pick the PPA, which we'd appreciate. You do it once, and you're done. Every time you shop, instead of going through Amazon, just go through the Amazon Smile part. It is everything exactly the same. It's all your information. It'll come right up. It costs you absolutely nothing, but we get a percentage, and that is from Amazon. So you might want to look into that. On a personal note, with talking to people, in Dublin, I met David and Emma, and they were exactly like me. And I don't find that very often because I'm a little off the crazy chart. I'm very interesting. But to find two people like myself has been, it's a blessing. There aren't that many like me. I have found others, but the reason I'm bringing them up is because Sherry, David's wife, emailed me last night to say she's so excited about the conference. They can't be here, but they're watching. And we've stayed in touch for the last year since we've met. And I just think that's amazing. I have my good old friend Rhonda, who's here, that I have not seen for years, and disease buddy Sherry is in here. People I've known for years, and I hope that you feel part of the family. I hope you feel welcomed, and thanks for being here.
Shelley?
Oh, Shelley, you got to stay here for a second.
Shelley, you might want to come back.
Just hang on, hang on.
Hang on, Shelley. Just stay here. Just stay here, Shelley. We're bringing you out a zebra, Shelley. Yeah. Are you happy? Are you going to keep it in your backyard?
All right. Wow. Shelley, you've been with us for, when did you start? So far, you know, I remember her being like, "Oh, how do you do this in Excel? What is Excel?" Something like that. And then, you know, and I was fresh out of the pharma industry and with all these like financial control mechanisms and stuff. And, you know, we talked and we sorted it out. And I subsequently forgot all of the subtleties, and then she now reminds me about all of the subtleties. But she's just taken the role to super high level. She's been such a great financial steward of the organization, and so we wanted to present this plaque. You're going to have to hold that.
I love it. That is amazing. Thank you. How am I going to get that home?
I don't know. We'll figure it out. To Shelley Schiffman, in appreciation of your years of selfless service as board member and treasurer of the Periodic Paralysis Association, you have bettered the lives of so many who struggle with this disorder. Love, all the PPA board.
Oh.
And wait, on the back, it's on the back too. All right, very good. All right, we'll figure out how to get it back to you. But thank you, yay. So next, we want. Oh my gosh. We had invited Patrick Cochran to the conference. He was the founder of the Periodic Paralysis Association, and we had wanted to give him a plaque. He founded the PPA for his wife Kay Cochran, who had Anderson-Tawil syndrome. And, she had passed away. He stepped down from the Periodic Paralysis Association, so he couldn't make it. But we wanted to, we made a, we made a how do you, like a memorial research grant. So like every year we want to give around $3,000 to upcoming researcher or someone who is contributing to the advancement of periodic paralysis. We do these things with the contributions that everyone's making, and hopefully we can find cures from this. So I don't know if you want to read it.
Yep. This is a certificate of appreciation, and it's awarded to Patrick Cochran, who's a PhD, and his wife Kay Cochran, who passed away last year from the effects of periodic paralysis. She had a heart-related problem. She had ATS. And it says, our organization would not exist without your tremendous dedication and vision in the field of periodic paralysis. On behalf of the PPA and the hundreds of members you have touched, we honor Kay this 25th day of October 2019 with the creation and awarding of the first Kay Cochran Memorial Research Grant to support research in the field of periodic paralysis.
So that was for Patrick. And the first recipient of that is Jack Tarleton. So he should come up. So congratulations. Thank you so much for all that you do. Jack is going to be our first presenter. Jeff, are you introducing or? I don't know. Do you have the? Okay, that would be great.
Jack, good to see you again. Silence. Is gold. Silence is golden. Yeah. Dr. Jack Tarleton received his PhD in molecular and cellular biology from the University of South Carolina. He's the retired director of the Fullerton Genetics Center Laboratory of Asheville, North Carolina, and served as director of the Molecular Genetics Lab section. While serving as lab director for over 20 years, he witnessed the explosive growth of genetic information, and as specific genes related to genetic disorders were identified, Dr. Tarleton was drawn to the work of understanding the molecular details of how genes function. Now, I was told at a previous conference that sometimes the blurbs that we're able to get the information about so that I can read them to you may not be all or inclusive. So, Jack, we're very happy to hear from you today and have you here today. And if there's anything that I've neglected to mention, please mention it.
Oh, great. So I don't know if you want to do the podium or the. Yeah. Oh, it's up there.
Wow, thank you very much. Is this on? Can you hear me? No? How about now? Okay. Thank you very much. It's an honor to be a participant here, and I came into an interest in periodic paralysis later in life, and I was about ready to retire, and then all of a sudden I have a new job and a fascination with this disorder that affects so many people, and we really want to make sure that when we were talking about genetic testing that we're looking at all the possibilities, and we've had a number of genes that have been isolated over the years, but there are a substantial number of people that don't have one of those mutations. And so I have focused on doing some work to look at patient samples that were collected mainly by Dr. Lehman Horn over the years. And when his lab closed down, we ended up taking those in our lab and just decided about 2 years ago that we should do a more formal investigation. So what I want to talk about today is some of that work, and it's definitely a work in progress. We're still trying to figure out what would be our goal would be what is the best approach in terms of genetic testing, what's the most cost effective, what's the most technically correct way of doing it, and that's what I'm trying to address today. So let me make sure. Okay. So I picked this slide with a cross-section of muscle tissue just to illustrate how compact everything is tightly packed in. So I'll talk a little bit about the known genes that we know about in terms of the ion channel genes that are located in the sarcolemma, the membrane that surrounds the muscle cells. And these microfibrils are in parallel with each other, and mitochondria will be mentioned today. We'll talk about the powerhouse that makes ATP in the cell and how that might be affecting some individuals. And just kind of the anatomy of the structure of the muscle cell. We'll talk about T-tubules, this network of membrane that goes down inside the cells. So I chose this just to illustrate how tightly packed these literally hundreds of proteins, maybe thousands of proteins are, and how they may affect each other. So just a little bit of background. Our laboratory in Asheville, North Carolina, has done testing for about 25 years, and I got interested in the ion channel maybe about 2005. And when Dr. Lehman Horn's laboratory started closing down, we ended up doing a lot of testing for the 4 genes that were known, the sodium channel, the calcium channel, and the potassium channel, KCNJ2, and a second calcium channel that's involved in thyrotoxic periodic paralysis, KCNJ18. So that was the standard panel for testing for a long time, and still is to some degree. The KCNJ18 testing has it's still performed, but it's not routinely done, I think, much anymore. And that's just because some of the results were a little hard to interpret. So I wanted to talk a little bit about and focus on the disorders that are not the ion channel disorders. Those that have the primary disorders, as I define them up here, is that the primary periodic paralyses are those that have a family history, typically, so they're familial. Sometimes they may be brand new mutations, and so they may still fit into the primary category, but they are not necessarily inherited. You can get a new mutation. The secondary causes, these are found in patients that have a disorder that's unrelated to ion channel. So no ion channel variant is present, and these examples of this may be renal disorders and hyperaldosteroidism, and the periodic process that's associated with hyperthyroidism. So those are secondary causes, but what I really want to focus on is the atypical ones. These are patients that have unusual clinical presentations. We do the panel testing, we don't find anything in those genes, and then we're kind of, in the past we've been kind of stuck, but there's been some recent advances that I wanted to talk a little bit about. So examples of these may be the disorders that are associated with the ryanodine receptor, the RYR1 gene, that in recent years we found more and more examples of. And then there's these so-called benign variants in SCN4A, the sodium channel gene, that sometimes get paired. By themselves, they seem like they would be benign when we do our workups, but they seem to be occasionally paired with another variant in a different gene, usually a chloride channel gene, that may actually give rise to symptoms. So about 2 years ago, we embarked on a somewhat of an informal study to look at some of the families that we had obtained samples on through Dr. Lehman Horn. And we did a technique that's called whole exome sequencing. So this is a look at not just a single genes, one gene at a time type of thing. We're looking at all 22,000 protein coding genes in the human genome. So part of the problem is that creates a lot of data, and the analysis of it is really intense and laborious. But it may be that this is the best approach for doing the types of testing that we need to, when we really don't know what gene we're looking for. This is probably the best approach. So we selected some patients and just contacted them, got informed consent for doing the study. So these are patients that are negative for the known pathogenic variants. And we used to do that with a technique called Sanger sequencing, which is a 1-by-1 gene. And we used the exome sequencing is what we call next-gen sequencing. So this may be a better approach because we're really on a treasure hunt looking for new alterations and trying to fit new things together. I will say that this is a very limited, it has a lot of limitations to doing this. The real proof of principle is to have functional studies. And so what we're identifying are genes that we might want to focus on, but eventually want to do functional studies on. And that's another level of complexity that we didn't address. So we don't have anything that we can say with absolute precision that we have found a particular gene. I do think we've found several situations that contribute to patients having periodic paralysis without having one of the known primary forms. Just a patient overview is that we contacted about 22 families. We had, most of this work was done in our lab, but we had some work that was done in some of the commercial labs that we analyzed after they were done elsewhere. But typically the best way to do this is to obtain samples from multiple family members, somebody who's affected, and maybe even two people who are affected, but usually a parent and a child, or perhaps in some cases we used distantly related cousins that had similar symptoms, similar clinical symptoms, and we wanted to take the measure to find what they had in common with each other from a genetic point of view. All of these patients have a history of partial or full paralytic or severe weakness events that are episodic. Most of them carry a clinical diagnosis of hypokalemia, periodic paralysis. But that sometimes just means that there also could be normokalemic. They tend to respond, most of the patients tended to respond to oral potassium. Most people had the common triggers that you're familiar with. But these patients sometimes seem a little bit more complicated. They may have some other symptoms that I've listed out up here that may actually be contributing to the difficulty in diagnosing them. They're responsive to different medications. Sometimes that's a clue. Diamox would necessarily be effective in all cases. Once we identified what we call a gene variant, a change in the gene. So you'll hear the term mutation and variant used somewhat interchangeably. But typically when we say something's a mutation, we have fairly conclusive evidence that it's a pathogenic alteration. When we identify these variants, these are what I call candidates. We are looking to see, does this make sense? Do they fit physiologically with what we think is going on? So I'll refer to these more as variants than mutations, but sometimes they do cross over and become mutations once we obtain enough proof for deciding that they're really meaningful. So the known mechanisms that we know about to date physiologically is that the sodium and calcium channels that are involved in primary periodic paralysis have what's called a gating current. These alterations, these variants that occur, these mutations that occur in those genes alter the voltage sensing function of the channel, and that's called a gating current. So we know that the mutations cause a little bit of a leak. It's like a little short circuit that occurs there. And in the KCNJ2 gene that's associated with Anderson-Tawil, we know that that's kind of a loss of function of that gene. The gene product is not there in the amount that's a normal amount. It causes its effects related to losing function as opposed to the leaky channels that are kind of a gain of function in the protein. Over the course of the last 6 or 7 years, there have been about half a dozen publications in the medical literature that describe SCN4A variants that seemingly when we go through all of when we find these, they're not in the typical places that the other sodium channel variants are found. And we go through a lot of analytical process to look and see what are these really in a spot in the gene, or are they causing a functional problem that might impair the gene. Typically, by themselves, they seem not to be causing a problem. But occasionally they get paired up independently with an alteration in another gene, and typically in the cases that have been reported, that's been the chloride channel. The chloride channel is an ion channel as well, and the chlorine going in and out of the cell is involved in restoring the resting potential of the muscle membrane and enabling the next cycle for an action potential to come down through. So the gene that's involved in that is the CLCN1, the chloride channel gene, and you may know about that from myotonia congenita, the disorder that's associated with that particular gene. About 2013, there was a study coming from France, from a group there that looked at a number of individuals that had an alteration in the mitochondrial gene that's called ATP6, and another one that's called ATP8. And those two genes overlap. So mitochondria, you may know, are independent organelles inside of the cell, and those are involved in generating ATP. ATP powers a lot of things that go into the cell. A lot of the chemical reactions wouldn't work without ATP being involved. And the mitochondria in the typical cell, there may be hundreds of those independently circulating there or involved in the cell. And those generate the ATP for powering things, but they do it internally. So there is actually a piece of DNA, a circular piece of DNA inside of every mitochondria, and actually multiple copies of that. And there's 37 genes in the mitochondrial genome, as opposed to the human genome that has 22,000 that's located in the nucleus. So the mitochondrial genes are involved in these bioenergetic pathways, and if ATP production is reduced below a threshold, that actually can impact a lot of the biochemistry that's going on inside the cell. So in 2013, this group found a series of patients that they identified that had these mutations in the mitochondrial genes. And that's as opposed to genes in the nucleus that actually can affect mitochondria as well. And I'll talk a little bit more about that in a moment. About 3 or 4 years ago, the first variants in the RYR1 gene that I mentioned earlier became characterized. That gene has been well studied for a number of years, but these were patients that had episodic weakness, episodic paralytic attacks. They actually had an atypical clinical presentation compared to patients that have primary periodic paralysis. And it turns out that there's, because that's such a big gene, there's probably a fair number of people that had an undiagnosed case that do have RYR1 alterations. And so we've uncovered in our series of patients something like 30% of those patients have an RYR1 variant that we are suspicious of. We can't definitively say, but we think it probably is related. And then about a year ago, there was a publication about this time last year looking at the sodium-potassium pumps. These are proteins that are embedded in the muscle membrane that actually shuttle potassium and sodium in and out of the cell. And so they're cyclical, so they're pumping it in and pumping it out. So there's a gene called ATP1A2 that was reported, a patient was reported to have. It's a study that came from a group in Brazil and collaborating with a group in London that Dr. Hanna is involved in. And they identified one patient so far that has this alteration, the only one that I'm aware of. So those are the mechanisms that are known already. I borrowed this from this illustration from Dr. Cannon's, one of his papers from a couple of years ago, a commentary that he did. And I just wanted to point out, if you look on the left-hand side, that's the sodium channel. That's a cartoon of the sodium channel as it winds its way. So that ribbon is a string of amino acids, which is at the genetic level, that's the gene encodes the precise order of those amino acids, and they have certain biochemical properties. And the one on the left side of that slide is the sodium channel. The right is the calcium channel, two independent genes. But you can see how similar those channels are, and you can the notation there is looking at the voltage sensing portion of the gene of the channel and mutations that are in that. So we have a very good understanding, thanks to people like Dr. Cannon, for understanding the sodium channel and those leaky currents that occur there that give rise to primary periodic paralysis. And I wanted to say that I put this up just because that's well understood. The mitochondrial, the sodium-potassium pumps, the RYR1 variants involved in the ryanodine receptor gene, those are not very well understood. So we're kind of at the cusp now of moving into a greater understanding of kind of the broader spectrum of genetic disorders that give rise to periodic paralysis. So this is the paper I was mentioning from the group in France, and so the title there is in orange. So it's episodic weakness due to mitochondrial DNA mutations in the ATP6 and 8 gene. There's a lot of technical things that go on related to mitochondrial function that really haven't been well characterized. But these deleterious mutations occur in a substantial number, if not close to 100% of the chromosomes that are the mitochondrial chromosomes. And these patients had episodic weaknesses, but they were actually Diamox. Acetylcholine was actually very effective in treating these patients. And so there's only this report of the mitochondrial disorders that have been confirmed. I have yet to see a second follow-up on this. So this may be a rare cause, but I think it's something, the energetics that are involved in mitochondrial function are really powering a lot of things. So even the sodium-potassium pumps, they take a lot of ATP to actually work. So things that fall below some bioenergetic threshold may be impacted by a reduction in function, if not a complete loss of function in the mitochondria. So the paper about 2 or 3 years ago that came out related to RYR1, this was actually the second report of RYR1 having involvement in periodic paralysis. But the original paper, I think, came out in 2010. But the paper reported on 3 additional patients that had alterations, had variants in RYR1. So the cartoon that I've put there is just pointing out. So the calcium channel, so this is one of those T-tubules that I was mentioning earlier. So the calcium channel is here and goes through the sarcolemma membrane, but it actually is in contact with the ryanodine receptor. And this is the sarcoplasmic reticulum that is loaded with calcium, and calcium is really the driving ion for muscle contraction. So you can understand why if something that comes in contact with the calcium channel has some alteration in it, it may give you a clinical manifestation of something that's similar to the calcium channel phenotype that comes with mutations in that particular gene. So that makes sense that there would be some connection there with the rhianodon, and maybe there are other genes, products here that may be involved in the future if we, as we study this a little bit more, we may find some of these other alterations there. So this time last year, this ATP1A2 was reported. So this is, as I mentioned, was from a Brazilian group in collaboration with the group with Dr. Hanna's group in London. And so they found that the variant that was occurring in this particular protein was really related to having a leaky mechanism in a similar fashion, analogous fashion to the chloride channel, or the sodium channel and calcium channel. This patient actually had seizures as well, so an atypical clinical presentation. The protein that comes from the ATP1A2 gene actually is tissue specific and muscle, is expressed in muscle, and it's also expressed in neuronal tissue. So the seizures kind of make sense that are a part of that spectrum as well. So most of this adds up in a logical way. I put a lot of verbiage here in this slide because I wanted this, when it goes into the meeting notes, for there to be some context. But these are places along the spectrum of the action, muscle action potential that potentially could lead to, if there are defects in proteins in particular areas, these may lead to clinical manifestations. So at the neuromuscular junction where the motor neuron interacts with the muscle fibers, there's about 50 proteins or so that are involved in making a normally functioning neuromuscular junction. And so there may be places, and we know that myasthenic types of symptoms originate from some of the defects in these particular proteins. And that might be a place when we're looking for alternative atypical types of genetic alterations that may be giving rise to periodic paralysis. We should look closely at the neuromuscular junction. And I mentioned to you the primary forms that are coming from the chloride channel and the calcium channel and the sodium channel. And I mentioned that kind of in the second here. These muscle action potentials are dependent on those working in concert with each other. The calcium that's released during after the muscle potential interacts with proteins, structural proteins that are troponin and tropomyosin that are in the structural parts of those myofibrils. And there are no known causes for periodic paralysis related to those. And I'm speculating that there may be some possibility of that, and I want to tell you a little bit more about that in some of these other slides. So we did this whole exome sequencing for a number of patients, about 22 families total. Sometimes we only sequence one person, sometimes we sequenced two affected people just to see if they would match up in some fashion or form. And these are variants that were found there, but I just wanted to pick a couple out and just mention them to you. So you can see at the very first one, there's an RYR1 variant. There are a number of RYR1 variants scattered through here, as you can see. And I mentioned earlier that we had a pretty high percentage of those compared to other alterations. We found some patients that had nothing that we could identify as a primary cause, but had a cluster of variants that would be associated with those proteins in the neuromuscular junction. So that became kind of a suspicious avenue for us to pursue. And we looked somewhat closely at those. Here's another part of those patient, the series, and we found any number of variants that we then had to go and explore one by one. And these, just to summarize the study findings, is that we found 4 patients that had sodium channel SEN4A alterations that by themselves we would have judged to be probably non-causal in terms of causing the independently causing symptoms. And in some cases these were paired up with the chloride channel variants that I mentioned to you earlier. And because that had been reported before, I think we were able to come up with a somewhat of a nice story for those families. And at least we've given a pathway that we can kind of go down. We had 6 patients out of the 22 that had RYR1 variants that we are suspicious could be involved in the pathophysiology. We would call these as having, we describe them as having uncertain significance because we have yet to do the functional studies on them. So that would be the gold standard for establishing them. We didn't find any patients that had alterations in sodium potassium pumps. And then there were 4 patients that we did not have any strong candidates for. But we did have several patients that did have these unusual alterations, and I wanted to really label these as speculative. These are not definitive at all, but they're suspicious in terms of having alterations cluster in a particular place. So we had 2 patients that seemingly had a number of different alterations that potentially could be involved in disrupting the neuromuscular junction. And interestingly, 2 of these patients really responded well to medications that are known to cause the acetylcholine to linger and reinforces the medications actually are treatments for the myasthenic disorders. So these patients, although they had episodic paralysis and weaknesses, seem maybe to have something more along the myasthenia part of the spectrum. And these patients had dramatic improvements when they took mestinone and 3,4-DAP, the diaminopyridine, and it really reinforced our thinking that maybe there's something going on related to the neuromuscular junction. But again, these are speculative. We really have not established that at all. And the other thing that we found was a couple of patients that had mutations in filamin C, which is one of the structural proteins that's involved in the myofibril function. And these patients probably don't have periodic paralysis, but they do have severe attacks of weakness, and it may be mimicking periodic paralysis. And they have a pretty complex clinical presentation that includes some cardiomyopathy. And it made us think a little bit about these myofibril types of structural proteins. The myofibrillar myopathies are a group of disorders related to defects in those genes. And sometimes it may be that some of our patients that are showing symptoms that look similar to periodic paralysis may actually have something going on in those. And we did have 1 patient that had a caveolin-3 mutation that is a known pathogenic alteration. So that's a variant that didn't need particular workup. But he also had a defect in another gene called POLG, polymerase gamma. This is a gene that's involved in normal functioning for mitochondria. The mitochondria would, if it's being affected, might be contributing to the pathology that goes along with caveolin-3 defect that we found. So this patient actually had some episodes that looked very similar to periodic paralysis, and was wondering whether that's a true case or not. We found other patients that seem to have defects that would be affecting the nuclear encoded mitochondrial genes that affect mitochondrial function. And so those are candidates in terms of affecting or interacting with these other genes. So we did find one family that has an SCN4A sodium channel variant that we would typically label as being benign, but also had a thymidine phosphorylase deficiency or an alteration that we speculate might lead to a deficiency of this enzyme. And that, the TYMP gene is a gene when both copies of it, one that you inherit from mom, one that you inherit from dad, are affected. That gene can actually give rise to a mitochondrial disease that's involved in depletion of mitochondria. So again, these are all speculative. We don't have real evidence, but it's giving us some pathways to pursue, and that's really what we want to do in the upcoming years. And I think I've kind of mentioned these associations here. So in summary, these atypical periodic paralysis associations often come with a complex of symptoms that really don't fit nicely into the established primary forms. So we've got kind of the defining hallmark characteristic is these episodic events that we all are familiar with, but they may have other things going on that really contributes to a more complex presentation. The RYR1 disorders, I think, are really going to be established as a maybe an atypical form, but to go hand in hand with the primary forms in terms of what we do first when we do testing. And of course, the testing that's being offered through Strongbridge includes the RYR1 gene. And so just to give you an illustration of how complex that gene is, it's about 4 times larger than any of the other ones. So when we start to do the work of analyzing genes, we have to go through a lot more data when we look at RYR1. We really couldn't possibly have done that testing with the older techniques. It's such a large gene, it just is not cost effective to do. It would take months to get a single gene done. So the exome sequencing or targeted genomic sequencing, where we do, target maybe 200 or 500 genes in a single testing, would probably be the best way to go. So the genomic approach, I think, is supplanting the old 1-by-1 gene. One thing that we don't have a definitive handle on is what other genes for these disorders that seem like they may be, that they're not the primary forms or not the ones that we have a suspicion about at this point. Should we be adding a number of other genes to our panel to be looking and screening some of the other neuromuscular disorders that may be related? We don't have the data to support doing that just haphazardly, but I think most of the testing that's being done by Invitae and some of the other commercial companies will, they also have an option for including other neuromuscular disorders, and maybe something like the filament C that I mentioned earlier, these myofibrillar myopathies that may actually contribute to some misdiagnosis may be covered by those. We also need to have better recommendations for how we do the variant interpretation. So there's a lot of complexity there, and it would be nice if we had a working group that actually standardized that. We're getting a lot of commercial lab derived sequences that are available to patients, and patients really don't have an idea of how to analyze those, so they come looking for help, and there are not many of us around that have experience in looking at periodic paralysis genes, and so it would be nice if we had a standard way of approaching that in terms of taking data that's produced in a number of different settings, either in clinical diagnostic labs or in laboratories like Dante, which a lot of people have had their genome sequenced. It's great to get your genome sequenced for, a few $100s, but it's the data interpretation that really is the critical part, and that's much more difficult to do. And in clinical labs that do genomic testing, that testing typically would cost several $1,000s to actually get the analytical part done. And it would be nice if we had a targeted group that we could focus on to do that. And then kind of lastly, it would be interesting if we could take some of our cases that are atypical ones that we don't have an explanation for and have group discussions of those in case conferences. And I think that would be a noble goal to do in terms of something that maybe the PPA could help us organize. So I'm going to stop there, and I'll be glad to take any questions. I'm not quite sure how much time I have. How close are we on time? Okay. Just move on.
Oh, yeah, so if there's any questions. Yeah. If there are any questions from the audience.
Yes. So, okay.
I think you kind of touched on this a little bit towards the end, but I'm one of those atypical people. Sorry. I think you kind of touched on this a little bit at the end, but I'm one of the atypical people where they have not found a gene, but they've clinically diagnosed me with this. And I would really like to be able to volunteer myself for the research community, but I don't have any way to do that. So, I didn't know if there is anything set up anywhere where I can say, hey, take my DNA, hey, ask me these questions, and if there's anything set up like that anywhere where I can volunteer my information.
So the question is there a mechanism for being involved in the research side? And at this point, there's not. It would be nice if we had another laboratory that's actually doing some of the work as well. Our laboratory is in a hospital that for 125 years was a nonprofit. And then about 9 months ago, we got bought up by Hospital Corporation of America and became part of corporate America. And their bottom line does not include doing a lot of those studies. So we're kind of left with piecing things together in terms of that effort. So there's not really a concerted effort at this point. I fully agree that it would be something that we should try to. Maybe the PPA could actually take some initiative in that regard in helping organize that. We desperately need others that would be interested in doing the genomic types of sequence, looking at the atypical cases. And there's plenty of opportunity there for young researchers that are interested in making their mark. These would be golden opportunities, I think. But at this point, we really have limited possibilities for volunteering at this point. Oh my gosh. Okay.
What? No, there's more.
Got one more. Got one more back here.
Hi, I was at the 2008 conference, and both my children and my granddaughter were genetically tested by Dr. Lehman Horn. They have hyperkalemic periodic paralysis, but what he was telling me is he felt like they had a variant on it, and I was wondering, since you were collecting that you did have some of the blood from Dr. Lehman's Horn testing, would it be possible?
I am not hearing much.
Oh.
Could you?
Well, she was saying that do you hear this? That her blood was with Frank, and there was a question coming.
Both my children and my granddaughter were genetically tested with hyperkalemic periodic paralysis by Dr. Lehman Horn.
Right.
And at the time, I was talking to him, this was in 2008, I was talking to him about the different symptoms, and he felt like there was another variant, another defect on their gene. And I was wondering, since you had the blood and was from Dr. Lehman Horn, could you do more testing to see if there is something additional other than the hyperkalemic?
Right. I think those samples that were collected in Dr. Lehman's Horn's lab in Ulm are available for study if somebody really wanted something done that's different than what we've done already. We have not studied, we've not gone back and looked at each one of those, and there's probably something like close to 100 of those. So there's definitely some capacity for doing additional work. What we really would need would be just a personal contact to let us know that you have had some work done, and then there may be some follow-up that we may need to do. So if you would just contact me individually, that we could maybe have a conversation about that.
One more here.
So my mom and I both have the benign SCN4A and lung QT, and our triggers are mainly salt and sugar. So our doctors are pretty sure there's one other genetical defect involved, and they're trying to find it, but are not sure where to look. Can you might give some hints? I don't know. Do you know anything?
Jake, I'm not hearing good. Could you interpret that for me?
So there are SCN4A variants, and I'm sorry, just repeat.
So we have the benign SCN4A, we have long QT, and our main triggers are salt and sugar.
Okay, so long QT, salt, sugar triggers, SCN4A. Is there another, their physicians suspect there's a second hit. Do you have a sense of where to look?
Typically, the chloride channel would be the first place to look. And, just based on one or two patients that we've done in this study, we should probably look at some of these mitochondrial depletion disorders in terms of being a second hit that might be aggravating the SCN4A.
I think just maybe in the interest of time we move on. I'm not sure. We could do that. The global thing here is, you have symptoms, and then you say, does it matter what genetic test you have or not if you take therapy and you respond to therapy? But then you want to know what you have, and if you have a positive genetic test, does it imply that you should do therapy A or therapy B? And it's still kind of a trial and error, and it's kind of like, what comes first, the chicken or the egg? I mean, is that? So we're kind of stuck in a little bit of a. At least we know for sure there are some definite genes that's our starting point. But now Jack is introducing ideas that, well, there's a whole lot of other proteins that we could look at, that if we have enough folks that are without the routine genes, you look at these other genes, and if the symptoms and those genes match up, at some point you say, you know what, that is definitely real and not just random. But it's hard to get a definitive answer, up front. I mean, is that fair?
I think with these atypical disorders that we're kind of blazing some new trails in terms of trying to understand them. There are any number of genes that we could actually think about adding to the targeted list, and just what I would like to see would be some standardization in the thinking process about that.
So I'll just make a comment. This is Steve Cannon. So it's a two-step process. One, for the atypical cases, it's complex genetics, as you've pointed out here. And when you're talking about novel genes that have not previously been firmly associated with periodic paralysis, that's an extraordinarily difficult nut to crack. And you're right about the approaches. I should let everyone else know who's frustrated that they've gotten back reports with variants in conventional genes like SCN4A or CNA1S, that In many cases, you can make an informed approximation and guess what the implication of one of these point mutations will have. But for one of us to study it in the lab, it takes one person 3 months at 100% effort to test one mutation. So you're not going to have high throughput functional testing of ion channels. It's just a difficult problem.
That's a great illustration of the difficulty that's involved in kind of bridging the gap between an observation and a genetic variant and getting to a agreement that that is actually pathological. And that's the dilemma that we have right now.
Now, there was a Nobel Prize awarded recently for oxygen and how it responds in different environments within the cell. Is there any part of that research that might help with, how calcium and chloride and other things that are affecting us? Is there anything within that research that you've seen that might help with this? They're pointing at each other.
Again, I don't want to monopolize Jack's time, but an interesting development in the field is thinking about the consequences of having these conventional periodic paralysis-associated mutations sitting there in the background all the time, but what brings out the episodic attacks? And I know the focus has historically been on potassium, which is appropriate, but we're learning it's much richer than that. I'll talk a little bit about this tomorrow. So it's not surprising that there might be many other systems, not only oxidative stress and oxygen sensing and things like that, because muscle is an amazingly adaptive tissue because the metabolic demands and the power changes radically. And so I'm sure some of these things will be tied in, but it's complicated.
I think what we're going to do now, if possible, we'll take a break for like 10 minutes. Bio break, bathrooms are over there. Chat amongst yourselves, and we'll be back in like 10, 15 minutes. Thank you, Jack.
Amanhã me socorreu com flores e aves, suaves soltas em asa azul, borboletas em bando. claridade de um novo dia não havia sem você. Você passou, me esqueci o que ia dizer. O que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete tocando blues. Que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete inventando blues. Que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete delirando no blues. Diz que pedra não fala, e dirá se falasse. Eu ama-me, queima na sua cama. O veneno da fala, esse beijo que é doce. Me prende-me, iguala-me, vende com sua bala. Diz que pedra não fala, e dirá se falasse. Eu ama-me, queima na sua cama. O veneno da fala, esse beijo que é doce. Me prende-me, iguala-me, vende com sua bala. Se desfaço de Zeus, te juro na deusa azul. Se me comover, eu já sei que é tu. Claridade de um novo dia não havia sem você. Você passou, me esqueci o que ia dizer. O que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete tocando blues. Que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete inventando blues. Que há pra falar? Onde leva essa ladeira? Que tristes terras vencerá? O intérprete delirando
the MC? You guys are the important ones. Okay, thank you.
And then Todd, can you come up on stage just for the video, just to capture? But then you're going to be walking around probably.
Okay. Yeah. I still, I was assured that I needed to do no prep for this.
Correct.
So I followed the instructions to the letter, and I have done no prep.
It's just asking people if they want to share their experience, their story, one aspect.
Basically, you just need somebody to be able to hold a mic around.
Yeah, but then they're going to say stuff, and then you're going to comment on it a little bit, and don't hog the mic. Try to get as many things as possible, but obviously they're going to say things that you really need to comment on, and just comment. Okay. I think Cannon will jump in if there's things, and I'll jump in.
Cool.
Lewis will jump in. He's over there.
I just wanted to tell you, I really enjoyed hearing you back in 15 when we were over. You came in 2015, didn't you?
I can't remember when it was. It was in Arizona. Whenever it was Arizona.
It was awesome.
Yeah, so it was actually someone else that you enjoyed hearing in 15. That's what you're thinking.
But he
of,
looked
right.
like you.
Right, yeah, but he was really handsome.
No, but I did. It was awesome stuff. Thank you very much. Okay. And I'm almost ready.
No, I'm just trying. So that is, you want to go.
Okay, it's going to be short and sweet. Testing, 1, 2, 3. Testing, 1, 2, 3. Well, I want to hand the baton because, you want to. Well, no, I want to.
Hey guys, we're gonna start again. If we could get back to our seats, ish.
That sound good?
That's perfectly fine.
What I did want to mention as a follow-up to the last presentation is there's some low-hanging fruit. How many people are the only person in their family to have periodic paralysis? So there's like a bunch of, quote, index patients that are, maybe spontaneous mutations. I thought I was a spontaneous mutation for 20 years. And finally, I convinced my, mom and my sister to get tested, and then they tested positive and explained a whole lot of unexplained symptoms that they were having their whole life. So it's low-hanging fruit. If you're the only one in your family, provided it doesn't cause major, family. I mean, there's a lot of things with genetic testing. You can't just go and test. There's a counseling and everything. But it may be wise to, think about having that discussion with all genetically related family members to see who might have it, because you can prevent a lot of bad feelings, with appropriate medication if indeed they end up having it. All right, Jeff.
Thank you. Okay, our next speaker, Dr. Todd Bell, is a pediatrician in the Amarillo, Texas area and has over 18 years of experience. He attended the University of Arkansas College of Medicine and graduated in 2001. He's affiliated with Baptist St. Anthony's Hospital and Texas Tech University Health Sciences Center, both in Amarillo. Dr. Bell sees many children with periodic paralysis and is the golden standard of care for these young patients. Dr. Bell.
I'm confident that no one actually put I was the gold standard for care of these patients.
It's the gold standard.
It's right. Okay. They assured me that I didn't need to do anything to prep for this, and so I followed their instructions. And so this is wide open. I would like, if it's okay, I'm going to steal the mic for about 90 seconds. And one, just explain why I'm up here. Mainly, they just needed somebody to carry a microphone around, is my understanding, and I'm able to do that. And so I'm going to fulfill that role. I actually got into periodic paralysis because of those atypical patients that Dr. Tarleton was mentioning. And that's actually the majority of the patients that I end up seeing who have paralysis events. And so, again, some of those other symptoms will be something that we may talk about tomorrow, I think, because I'm going to have another opportunity tomorrow to speak. The other thing that I was going to do is just agree with Dr. Tarleton in that there is definitely a need for developing some sort of research network, I think, and I think that that's something from the standpoint of the PPA and perhaps beholden on us as physicians as well to find a way to be able to drive that forward. with that, what am I supposed to do now? Dr. Levitt?
The idea is this is just a chance for everyone in the audience to, to share your story or some aspect of your story, because it's important for all of us to hear each other's stories, challenges, questions. It's kind of open time. I found this to be the most essential part of every conference that I've ever been at. And then, all the doctors in the room can comment.
All right, and with that, we'll start right here.
So I was diagnosed about 10 years ago, and then just recently we found out that it is in my family. My sister, who's here with her daughter, has it, and her daughter has it, and both of my children have it, but they're older. They're 20 and 25. So now that we are aware that it runs in our family, it appears to be part from my mom, part from my dad. It's the SCN sodium channel variant with the chloride channel. My dad had the chloride channel. All of the kids are in their 20s. My niece, who is here, she seems to be the most symptomatic. My children, my daughter seems to be a little bit more symptomatic than my son. So the girls tend to be a little bit more symptomatic. We do know that food is a big trigger, stress, all of those things. But now that we know what it is, how can we help our children moving forward? We are doing the treatments with them with the potassium and stuff like that, but my physical abilities have been severely limited from when I was younger, and I really want to try to prevent that for my children. And I don't know if the potassium they're taking every day, the diet that they're doing, is there something else we should be doing to keep them from getting the muscle weakness and that type of thing?
So we should also put a disclaimer that, as far as I'm aware, there is virtually no pediatric data in the literature. Again, if anybody else wants to weigh in on that, there's nothing. That being said, I think, first of all, very common questions and very good questions. And I think that there's a lot of things that we need to, it again highlights the need to really move forward with some of those clinical type studies to answer questions that we all have. What does it do to the body when you have a child who's on a high dose of potassium for long periods of time? We just honestly don't know. I think that, at least for myself, and we'll certainly see if anybody else wants to weigh in on this, when we're thinking about prevention of problems down the road, I don't know of anything specific to the ion channel disorder itself. However, there's this whole cohort of other symptoms that also seem to be associated as well. For example, a lot of folks will have issues with hypermobility. Dr. Tarleton mentioned hypermobility. We know that with hypermobility, you have an increased risk of joint dislocation. And if you use Ehlers-Danlos hypermobile type as sort of a model, or benign joint hypermobility syndrome as a model of that, what we do see is that once you injure a joint once, then oftentimes you'll have recurrent injury of that joint. And so being aggressive, for example, like with physical therapy, trying to rehab joints, maybe something that specifically when you're thinking about long-term function, musculoskeletal function, that may be something that is useful. We tend to be, in my hypermobility clinic, we tend to be really aggressive with rehab if it's a rehabable joint. There's some that aren't, like fingers, next to impossible to be able to strengthen the fingers. You got to really look at like ring splints if someone's having finger dislocations, or ankles are kind of interesting. So the ankles, we do try to do physical therapy. There are specifically some things they can do. I think the PT usually starts working with the, for example, the anterior tib, try to be able to improve the arch, try to improve or decrease some of the inturning of the ankle when we're walking, which causes a little bit of stretching. And and by doing that oftentimes can be helpful, but it's one of those joints that sometimes we also have to do bracing to be able to prevent problems down the road. I do think there's a lot of folks who have adult patients who have PP, or I suspect that there are a lot of adult patients with PP who actually have long-term functional issues that are primarily probably not due to their sodium channel issue or their calcium channel issue, but rather due to just that unrelated hypermobility that causes that wear and tear on the joint over time.
Oh, so, okay, question though. No, over back here. I'm back here. Turn 180 degrees. This is
like
God.
I'm on the road to Damascus here.
I think, the question might be, though, you have a child, they're 5 years old, you have a genetic diagnosis, they have a genetic diagnosis. Maybe their symptoms aren't that they're flat out paralyzed. So, I wasn't flat out paralyzed till I was 12. But I remember eating Lucky Charms and candy, going outside and feeling like I just feel like garbage when I was like 5 or 6, and I just assumed that, everyone gets the sugar crash. I'm sure that was, had I taken potassium, I would have been like, So like, when do you give the kid potassium? When do you give the kid Diamox? And how much?
Again, great question. Well, and so this is just purely what I typically do, is if the child is symptomatic to the point that it seems to be affecting their activities, mom's noting that little Johnny just never wants to go run play at recess, or the teacher has noted that. If it's something that seems to be affecting those activities of daily living, then I think it's probably worth starting to intervene on that. And some of those things, like throwing away the Lucky Charms, or even better, sending them to your pediatrician because he doesn't get paid much and he likes Lucky Charms, would be very a reasonable place to start, because obviously no kid is going to be harmed by a Lucky Charm deficiency. So some of those behavioral and dietary things, I think that's a great thing to do even if a child's not symptomatic. But if we're starting to think about using the medications, I think it really has to be, are we seeing any effect on those activities of daily living? And I don't know of a great test for that.
Are you asking the child, like, honey, are you tired? And how... When do you let up and when do you...
Right. Well, and that's part of the struggle, is because, again, kids tend to be fairly susceptible. So if I have when my kids were smaller, now I have teenagers now and they're not susceptible to anything I say, but when they were younger, I could make a suggestion and they'd, I'm tired, or, it's, sweetheart, it's 8 o'clock, aren't you feeling a little sleepy? And they're like, yeah, okay, I guess so. So I do think you have to be cautious with that. But certainly if it's reached the point that the parent has noticed or suspected or is, and I also totally realize that as parents, we also have a tendency to be real intense in our observation about our kids when there's something that we're watching for. But I think if the parents have noticed something, or at least they have a suspicion that there's some of those effect on activities, then maybe it's worth intervening and trying to intervene. Again, part of our issue is it's that balance. We don't want kids to be symptomatic, but we don't have great data in children on really any of these medications that I'm aware of.
So since we started, this is my niece here, since we started, now she started, just like what you were saying, her and my daughter, my son does not take it on as a need, as needed. My niece and my daughter do, and they say that it has changed their life. But they should just keep doing it as needed, right? Not on a daily basis. Like me and my sister take it every single day to prevent an attack, where with them, they're okay right now doing it as needed, right? Or should they be doing it?
It's going to be a trial and error kind of thing.
I think it is. I think the one thing similar to little Jake's Lucky Charms experience, he may not... or he didn't realize and didn't know that was different until he threw away the Lucky Charms or started taking potassium. So I think one of the struggles is, it is trial and error, because one of the struggles is you probably didn't know how good you were going to feel until you took the potassium. And it's like, wow, where did all that, where did the energy come from? How is it that I'm able to get up and go now and I couldn't yesterday? when the same thing happened. And so I don't know. There may be a role in trying to schedule things and see if there's a noticeable difference. I would suggest to you that if you can't find a difference, if you start taking it on a daily basis and you're like, man, I'm not sure that that's doing much, then I would probably back off and go back to the PRN, because as far as I'm aware, there's not any data, at least from a pediatric standpoint, that putting our meds in on a regular basis is gonna prevent an issue down the road. So it's really just kind of, is it going to be able to help with the symptoms and trial and error?
And we got over here.
Hi, I'm Miriam Breslauer. I'm 42 now, but when I was a child, I didn't really have full body paralysis, but I'd have random acts of weakness, or especially when I was doing sports, I'd be running down the basketball court and all of a sudden I'd feel like I'd run into a wall. And nobody around me, but just all of a sudden stop, and then I'd have to crawl off the court in the middle of a game on a regular basis, pretty much every time I played. And my coach just told me that I needed to lose weight, and I wasn't active enough, or not practicing enough. But over time, my skills in basketball, for example, got much worse. Like I couldn't shoot as far, I couldn't shoot as accurately, even though I was practicing as hard as I could. And it's just an example. I don't know if other people might have that as warning signs that periodic paralysis might be a problem, or might be a sign that there's something wrong. But people kept telling me that it wasn't abnormal. But for me, it was like nobody else is going through that.
Yeah. Well, first of all, I don't have periodic paralysis, but I also suck at basketball. So that may be that's probably irrelevant. But you do raise a very good point, and I think that is that one thing as your general pediatrician, if you tell your general pediatrician, hey, I'm concerned that my child may have periodic paralysis, and they go and they Google it or they look it up in Nelson's, then they're going to come up with a single paragraph that says something on the order of, you have these paralytic events. And so they're going to say, well, has your child been paralyzed? No, they just seem tired or weak or having trouble, discoordination. And so I think that sometimes for those of us who aren't periodic paralysis experts, we may have a more difficult time realizing that actually can be a sign of what's going on. Now, we obviously know, certainly in the adult population, is not every event or episode is a full paralysis episode. And so I think that one of the things that we try to kind of sort out with the kids, and honestly we depend a lot on the parents and the family for this, but do they have episodic weakness that's not otherwise explained? And that episodic weakness certainly could be evidence that we have periodic paralysis that's there.
I will say, and there's someone over there, I will say that a lot of people just, they wake up in the morning and it takes them a half hour to get going, and they're tired, and they just assume that that's the way they are. Just like I assume that, well, after you eat a candy bar, of course, you get tired and then it goes away, instead of realizing, hey, maybe I should be under treatment. So I guess you have to pay attention to the symptoms, and that's also the family members that are undiagnosed. If they have that kind of thing, like there are entire families that wake up slow in the morning, and and only one person has the diagnosis, and they realize that everyone has that. And the question is, how aggressive do you treat, right? Like, should I be taking more potassium, or am I functional enough? Should I move from acetazolamide to Keveyis, and maybe I'm going to get a better response, or should I leave well enough alone? That's that's always tricky.
Agree. Here and then there.
And we'll go around and we'll get everybody.
With it primarily being skeletal muscle, that is where the ion channels have been identified, is there any crossover into whether skeletal muscle mutations can impact smooth muscle function in an area, kind of GI tract type thing?
I actually have an answer to that, or I have a suspicion on that. But does anybody else who's actually a geneticist and knows, or a neurologist knows about these things want to weigh in on this?
There's certain areas where both a voluntary muscle and smooth muscle are acting on the same system, like bladder control and things like that. So there are instances where a channel defect in striated skeletal muscle will affect function, but the big players, the sodium channel, the calcium channel, the chloride channel, are not expressed in smooth muscle.
I think that my only thing, and appreciate that, my only thing I would potentially add to that is if you're concerned about, like with gastroparesis or small intestinal bacterial overgrowth or functional constipation, functional diarrhea, those type things, those are things that are all controlled by the autonomic system, and we're going back sort of towards the atypical a little bit here, but the difficulties in regulating the autonomic nervous system have been associated with certain hypermobility syndromes. And we don't understand why there either. But certainly autonomic dysregulation, and again I would use EDS hypermobile type as sort of a model of what that looks like, but all of those things in the GI tract do have autonomic control, so it's not a direct relationship to the ion channels that are involved with PP, but maybe something else that's related, just because it seems like at least that hypermobility is more common in PP population.
And we'll take over.
Right here.
Oh, yes. Sorry. Wow. I totally need to get my glasses changed.
We just barely got diagnosis for my husband. He's very atypical. His potassium levels are always normal, but he responds as a hypo, so potassium supplements help him. And we have a daughter that's like extremely atypical. She has been diagnosed with an autonomic nervous system disorder, and since she was like 1, she passes out often. When she got to be 12, she started passing out much more, which is a normal thing for young teenage girls. When it was the worst, it was 10 times a day. But she also started having the full body muscle weakness, more likely happening after a passing out episode. Could there be a link between the passing out triggering?
So the great thing about yes, no questions is I can say yes. So there is actually, and again, I certainly don't want to hijack from there's some science that we know because of a lot of the great work that's been done by some of the folks in the room here even. There's a lot that science just hadn't caught up with what's going on. There is at least a biologic plausibility that's associated there. Again, that autonomic regulation, the association with hypermobility, this clarified relationship of hypermobility with hypo or hyperkalemic or with ion channelopathies. But with the autonomic system, the most common type of dysautonomia in children is actually going to be a hyperadrenergic dysautonomia. So basically that's if you have the sympathetic nervous system, which is the fight or flight response, and then you have the parasympathetic, which is kind of the veg out and chill. And so the sympathetic is if a bear walks into the room, then I'm going to get this big uptick in my sympathetic system, because I need to be able to run away from a bear. With the parasympathetic, it's kind of what happens on Thanksgiving afternoon when you're sitting on the couch pretending to watch television, but really you're just trying to stay awake. And so it's the balance between those two that affect a lot of different organs. Well, some of those organs that are affected include the adrenal, the hypothalamic pituitary adrenal axis. That's part of that autonomic system. And we do know that when we get that adrenaline, it causes a lot of different things, but one of the things that it does is that part of what it does is it actually shifts potassium back into the cell. So the same sort of reason why sometimes we'll have patients with PP who will trigger because of a startle, that presumably that may be the same thing, same sequence of events that's happening there. So with the hyperadrenergic, so too much of that sympathetic nervous system, like with POTS, postural orthostatic tachycardia syndrome may have been the diagnosis that that,
Well, she doesn't fit very well in that bucket either, because
Okay.
she can be sitting still, not doing anything, or having a conversation and pass out.
So I wonder whether, one of the things that we kind of have thought about or speculated on a little bit is that if you have a kid who's predisposed to the autonomic regulation issues, so you end up with flares of the sympathetic nervous system. Those flares in the sympathetic system then cause the release of the cortisol and that those adrenal axis hormones, which in turn are going to shift some potassium into the cell, thereby dropping the, just like if you're giving somebody insulin or a donut, then that may actually trigger events. I had one little boy who has adrenal insufficiency that we finally had figured out that what the deal was every morning and afternoon, and it didn't happen every time, but he'd take his replacement hydrocortisone that he had to have for his adrenal insufficiency. But if he, when he did that, then we had this little drop in the potassium. He dropped down to about 3.2, and that was enough to have him go into paralysis. So we were able, in his case, just to be able to schedule potassium at the same time that he was taking his daily medicines for his adrenal insufficiency, and that was able to at least decrease the frequency of events. But it is tough. The hyperadrenergic dysautonomia that we see in kids typically gets worse as they start to go through puberty. So the ages 11, 12, 13, usually it typically kind of evens off a little bit after that, and a lot of kids will actually grow out of the autonomic issues. And then by the time they're in their 20s, that's gone, even though obviously the periodic paralysis may still be there. But if the stress overreaction, the adrenergic dysautonomia is a trigger for the paralysis event, that at least may get better as she gets older. Now that doesn't get you through high school, but at least there's a light way down at the end of a very, very long tunnel.
Okay.
I now have
We got someone over here.
How'd you get another microphone?
I got connections.
Yes, he
Jake's
does.
got like 6 microphones.
He's
Just
El
like stuck in his
Presidente.
pocket.
He can do anything he wants. I mean,
I guess.
so. Obviously, I'm in my 30s. Growing up, it showed up. I don't know, with you being a pediatrician, I went through years. We don't know what it is, but also too in the limelight. Are you seeing kids or even young adults dealing with periodic paralysis and how they're managing with ADHD or anything like that, or anxiety? I know, like when I have anxiety attacks, I burn through myself so quickly that I'm crashed within minutes afterwards.
Yeah, so I think great question, and everybody's pointing to people that they know.
Before you answer that, I also too am noticing, like I've just recently diagnosed autistic, so I'm sensory overload again, also causing major crashes too.
Absolutely. So again, the disclaimer that we've got no real data on this. When you're thinking about anxiety, and especially if you're thinking about panic attacks, and someone who's a neurologist is gonna laugh at me when I try to describe this, but basically, as you think about the portion of the brain, the neuronal circuits that are actually processing emotions, and how that communicates what those emotions are to the rest of the body. So what we feel as anxiety is not actually something that just goes on in the head. It's how our body responds to that sensation or that emotional processing that we then label as anxiety. So if I gave a medicine to, if I give somebody epinephrine, which is going to be something that kind of jacks up that sympathetic nervous system, they would at least potentially feel anxious. So again, I don't know that it's something that it's a direct ion channel issue, so that it's not necessarily directly related to the periodic paralysis, but there has been a couple of studies, and actually a nice little study that was, I can't remember where this was done, within just actually the past 4 years, that they went through at a tertiary psychiatric department, and they went through and they basically did what we call a Baton score, which is a way of checking to see if someone is really flexible in their patients who had generalized anxiety disorder. And about 80% of patients who have a generalized anxiety disorder actually are hypermobile. And if we think about what is hypermobility, that's a small 10% of the population. Biology is a bell curve, not named after me. And you have 10% there that would be more hypermobile than everybody else, just like 10% of people are taller than everyone else. Well, 80% of the people who have anxiety are in that 10% of the population that's hypermobile. So again, the thought then is, all right, is what we're seeing as anxiety or panic attacks, are those actually an autonomic issue, an autonomic regulation issue, which as you start to think about, PTSD or some of the work that's been done on use of beta blockers, treating the way that we treat performance anxiety. If someone's getting up in front of a group and is going to be speaking and they're really nervous and shaky, what do? We give them something that dampens that fight or flight response. And lo and behold, they don't feel as anxious. So I do think there's probably a link. There's no direct link from an ion channel standpoint that I'm aware of. But again, we have this whole list of ancillary symptoms that are not explainable, in my mind anyways, directly from that ion channel, at least the ones that we know of, as far as genetic mutations, but it seems to be a recurring theme.
But you can definitely see a feedback loop. If you're anxious and you have adrenaline, and adrenaline is a cause of...
Are you like under a table?
As a trigger,
no,
it
you're standing right beside him. I'm sorry.
It cycles through.
Is there any chance you could get like one of those little orange triangle flags or something and just sort of attach it so I can see you? I'm having trouble here.
Yeah. I gotta... Yeah. Someone else.
I think we had one here.
Okay.
We'll go...
So this is kind of a long question, but it's all part of the same question, I promise. So we're hypo. Myself and my son and daughter have it, clinically diagnosed, and so we take a lot of potassium, and the potassium works. But we had a doctor ask us where the potassium goes, and I tried to tell him it goes in the muscles, but he said, but it doesn't stay there forever. And so I'm thinking that we pee it out when we're not in an attack, maybe. These large amounts we're taking. So the second part of the same question is, I have reduced kidney function by quite a bit, and I'm wondering, are we damaging our kidneys by taking this potassium, and is it safe? I mean, obviously we have to take it, but I'm wondering, is that what's going on here? Also, I have symptoms of, tea-colored urine sometimes, and a lot of times on the same days that I'm very achy, so I was wondering if that would be possibly rhabdomyolysis. Myolysis?
Sure. Yeah. So the potassium does, we think that the small fluctuations of the potassium are probably intracellular shifts, and then it does come back out, and then the body pees it out. And there is some data that the kidneys accommodate to chronic large potassium dosing. So which is part of why we sometimes have difficulty depending just on potassium to be able to manage symptoms, is because over time folks get better. Their kidneys are just really good at peeing out potassium, and that's where some of these other medications I think sometimes come into play. I don't know of any data as far as I'm aware, there's no data to say that taking the potassium as a child is going to cause kidney issues later on. But again, there's just not a lot of data that's out there from that standpoint. And with tea-colored urine, certainly rhabdo would be one thing we would think about. I don't know. There's so many things that can cause issues with the kidney. I don't know that I would necessarily say that it's likely that the reduced renal function that you have is necessarily because of prior potassium use. Even like the rhabdo itself, if because there's other things that can also predispose to rhabdomyolysis. And someone who has had rhabdo, that can actually cause kidney damage as well. Anybody else want to weigh in on that one? Jake, is he back under a table?
Yeah, I'm back here. I changed position. I'm here. I think anecdotally, so you have elevated kidney function? Or
reduced.
Reduced.
Kidney function. Yeah.
So I mean, I anecdotally, if you take the whole population of periodic paralysis, I haven't heard. We take a lot of potassium. I haven't heard of renal function being reduced on that basis. Rhabdo will mess up renal function if you have recurrent serious bouts of that without rehydrating and stuff. So maybe that is a thing, but more of a nephrologist.
Would the rhabdo be
I guess that's like a heavy word, the rhabdo, but there is some muscle breakdown that happens when you have attacks, and if that happens on a massive scale, then yeah, I wouldn't call that as a definitive, and it's something that you'd have to kind of like notice what it is, do the blood tests, and make sure there's a temporal correlation, and if it happens twice and 3 times, then you start correlating it, but it would be like a one-off. You have a question?
Yes, my daughter's 13, and she started showing symptoms at 1, but wasn't diagnosed until 4. Neither myself or my husband have periodic paralysis, and we've been genetically tested, all 3 of us, and my daughter's been published in the medical journals. We've noticed, as of the last year and a half, a decline in her health overall, in her ability to walk. So she went from being able to walk last year to not being able to walk, and she's been in this wheelchair now for probably about a year and a half. And I guess as a pediatric doctor who sees patients with these conditions, what would you say or recommend? I mean, we've seen things that do kind of help her overlying condition as far as episodes go. She's on liquid potassium and Cavis, but I guess the physical ability to walk has kind of been our biggest concern, losing, just her youth, I guess.
Sure. Anytime that we have a kiddo in a wheelchair, that when we want them to be out kicking soccer balls, obviously that gets us. Just to kind of clarify, so the with the HPP does she have one of those 4 common genes, or do you know what the genetic variant was, or?
The variant was R897S.
Which channel? Sodium, calcium, potassium?
I think it's the calcium.
Okay. So the I guess one thing that I would just mention or think about is with a decline in function in spite of therapy, I would actually, and so she's being treated, and you guys are on that, and she has this progressive decline. I would also be just scratching my head about, could there be something else unrelated that's going on? Now, obviously, you've got some great docs who've been taking care of her, and I'm certainly not second guessing them. I will say, as physicians, we have a tendency sometimes when we get ourselves into a little bit of a rut, once we know what we think we know what's going on, it's hard sometimes to be able to look outside of that one pathway, that diagnosis that we've already put into place. And progressive weakness, I'm not familiar with that particular variant. I don't have any other kids who have that variant. And so it may be something specific to that particular calcium channel ion issue. But I would also at least keep my eyes open. Could there be something else that's also going on there?
Well, I guess over the years she's had a lot of,
We've studied 897S in the lab. It's the most severe leak of any hypo-PP mutation we've ever seen. So unfortunately, there's a pretty rational explanation for why your daughter's symptoms are coming at a much earlier age than would usually happen in hypo-PP, and she's much more profoundly affected. I'd love to talk with you at this meeting about it. Okay?
His answer is more definitive than mine. Sorry.
Yeah. Oh, I know you. We're going back and forth in the room, so you go someone down there, I'll go someone down here.
You mentioned you had a patient with Addison's and periodic paralysis. I'm a pediatrician, and my patient is actually my wife, who has Addison's and periodic paralysis. She runs into a problem where she needs her sodium for her Addison's, but she takes too much sodium, it affects her periodic paralysis. Where do you get the balance?
Be turning that one to Dr. Cannon as well. I don't know. I think it's a trial and error. I do think that at least with this one kiddo, the one thing that we did that was actually really helpful with this particular kid was timing some potassium when he was going to be taking his hydrocortisone. And we end up writing up, you know how it is with adrenal insufficiency. Everybody's got a letter that you take. If you have to go to the ER or if you cut off your leg with a chainsaw, then whoever is supposed to read that letter and say, hey, this is what I'm supposed to do. We're going to do whatever your doc's doing, 3 times the usual dose or whatever it is. And so we just actually basically just hijacked that and then added in that, you know, with the standard dosing, he was going to take X amount of potassium. With a stress event, if he's getting stress dose, we're going to do this other amount. That may be worthwhile. It is, I think, though, really difficult, in part because there's so much variability in physiology just from person to person. Not that the physiology is different, but the magnitude of any physiologic process varies from person to person because of genetic variability and body mass and all those different things that happen. So good luck with that.
We have over here.
Hi. I come from a large family where there are about 30 people who have some sort of symptoms of PP and muscle weakness. We are yet as to be diagnosed or have a conclusive gene defect. But one thing that I found, it seems there's a suspicion that the symptoms can get worse for each generation. And I've never stumbled upon that possible fact in any literature. So my question is there any evidence at all that PP can get worse for each generation?
And I think it's a wonderful question for us to ask our geneticists. Do we have geneticists? I'm not aware of any data to say that we have that. And there definitely are genetic diseases that we see that as a pediatrician. Not for the life of me that I can think of one right now, but there are frequently, like with tandem repeat mutations, like with is it Huntington's maybe that you get worsening over time.
Dermatology, I'm sorry.
Yeah, okay, all right, yeah. We're of no use to you on that.
Steve's comment.
These are the trinucleotide repeat expansion diseases that get worse with successive generations. So there are some that affect muscle. Myotonic dystrophy is the biggest one. It's the most common form of muscular dystrophy. If your family has not been screened for myotonic dystrophy, you would certainly want that to occur. But for the conventional forms of periodic paralysis with the ion channel mutations, there's no progressive worsening with successive generations that I'm aware of.
I actually do know something about myotonic dystrophy. I should have been able to answer that. Yeah. Right. My turn? Did we have anybody? Yeah. Oh, we're right over here. Yeah, I handed my mic away. I was really hoping you were going to actually answer that question earlier when I handed you the mic.
He's going to pass it off to wifey. I was clinically diagnosed hypo in 2012, and then last year tested positive for RY-R1 variant, and then our daughter also tested positive a few months ago and has been showing signs. She's 8, yes, she's beautiful, wonderful, we love her just the way she is. But she tested positive, and we've seen some signs. And her pediatric neurologist is very kind, had never heard of the disease before. So she's open, but she does not want to put her on potassium supplements because she tested normal, in the normal range. And I've said her mama also usually tests in the normal range, but needs it. So I guess I would love some advice as far as how to talk to her doctor. I am not a medical doctor, don't claim to be, but I do have a mama's gut and suffer the disease myself. And we try to avoid triggers as much as we possibly can. That's how we've been managing it thus far. But I would really love that option if it's something that is supported in the community. My husband's whispering to me, maybe he's going to say, maybe I should mention, she's also on the autism spectrum and has ADHD. So.
Yeah, so I think, going back to kind of like we were talking earlier, I feel pretty strongly that if a kid if they're symptomatic to where it's affecting their activities. I mean, you know, we can tell that the kid has symptoms. I really think that they need to be able to be treated, and at an absolute bare minimum, they need to be able to at least have access to the fast-acting potassium, right? And so because we got to have that emergency plan in place. How do you approach your doctor on that? It is difficult, I think, because we don't want to. Sometimes us doctors have kind of thin skin. We get our feelings hurt, and nobody really appreciates us. Also, sometimes as doctors, we need to just suck it up and realize that we don't know everything. I think that when you're approaching the physician, I think that it, in my mind anyways, if you can present what the issues are, what your concerns are, and then open it up to what do they think is the appropriate treatment. Then, and you can certainly, you know, after they do that, they say, well, I think we just need to watch and wait. Then you can say, but my concern is this, you know, what if we're in an event, she has her first big attack, and we go from these sort of subtle symptoms of, well, she seems a little weak on Tuesdays after soccer, to all of a sudden now she's laid out over on the side of the soccer field and can't move. What do we, you can just ask your doc, what do we do in that hypothetical situation? Because the answer is not, oh, well, we'll cross that bridge when we get there, because we've got to have a plan in place to be able to project what might happen down the road. And then, and I think that maybe if you can use that as a jumping off point to be able to say, you know, well, you can even, you know, say, Dr. Cannon said that you should have some sort of emergency potassium and or whatever. But again, at least let them have that opportunity to think through it. Because I will tell you that and then if you still aren't comfortable with that, even though your pediatric neurologist is a lovely person and it may be difficult to find another one, I think that arguably the most important thing you can get out of your physician is someone who's willing to listen to you and take your concerns seriously. And I think that if you're not getting that, if you're uncomfortable with that, and you've talked to them and you just don't think it's going to work, then I think you maybe need to look for a different pediatric neurologist. I will tell you, the medical system is designed for the 80% of patients who are average patients. Average from a disease process standpoint, average from a payer source standpoint, average from height, weight, location, distance from the physician. This system is unintentionally, but it's designed to accommodate that 80% of folks that are close to the median. And when you start looking at things like periodic paralysis, you're really outside of that 80%, and it becomes much more difficult to make this system work for you or work for the patient. And it almost seems like you're having to work against the system to get anything done. It is, I think, a really difficult position, especially if it's someone who's a well-meaning doctor. And I guess another potential thing would be reaching out to the PPA or to one of us and just to be able to talk through what the doctor's concerns are. So if your doc wants to give a call, and I think that probably, I'm sure Jake does a lot more than I do, or some of our others, but even I occasionally get phone calls that there's a doc who just wants to say, hey, I had this patient said that, you know, they want to do such and such. Is that completely crazy? And I'm like, no, that actually seems like a really good idea. And oftentimes that can also be reassuring to your physician as well.
Yeah, but we leverage PPA Ask the Experts that way, and if you can ask the question, or hopefully the doctor asks the question. That's like so rare, and then like I can connect the doctor to a neurologist or myself, and we can sort out those issues. My question, two questions. I guess you can get to a diagnosis of periodic paralysis by the hard genetic test, and you can get to it by clinical diagnosis. So for sure, you're clinically low potassium, you get better with potassium, you have bouts of weakness. So clinically, your periodic paralysis. I feel comfortable making, you know, recommendations if there's a clinical basis. The other thing, I guess, a question for Steve is so the RYR, is that like a thing now? Is that like a new? Should we be saying sodium, calcium, and RYR?
Jake, it's a thing. We're on a first name basis. So. In all seriousness, the challenge with the RYR story is it's one of the atypical variants of periodic paralysis. There aren't enough experience, isn't enough experience yet to know how responsive it's going to be to potassium. As you heard from Todd, it's potentially big bang for a small buck, right? Potassium is relatively safe if you have normal renal function. So it's certainly worth a try. But scientifically, we cannot connect the dots about why it should even be hypo versus something else. But clearly you've experienced that, and that's the most valuable thing. But the RYR one, I would predict they're also going to play out a little differently than some of the traditional voltage-gated ion channel ones that we heard of. So as I'm sure you've read and are aware of, problems with RYR1 can present in many different ways: malignant hyperthermia, central core disease, mini multi-core disease, all these other things. And just the overlap of those muscle syndromes is still an active area that's being worked out right now.
If I could just add something that I don't know whether it's helpful or not, but you know, there's a lot of really well-meaning doctors out there that actually just don't have the information. And so over time, I've just found our collective curiosity and the fact that we just love to read and we just never give up. If you haven't found the article that Jake wrote in 2008, which is practical aspects in the management of hypokalemic periodic paralysis, it's a great starting point for everybody, but it's a great starting point for a lot of doctors. Yeah. I mean, we live in Toronto. We've got a wonderful hospital there with a lot of unbelievably well-trained specialists. And when Matt was first diagnosed very, very early, we got a diagnosis very accurately and very early. But in terms of the actual management of the issue afterwards, it's a little bit of hit and miss. I think it's a lot of self-advocacy. It's a lot of trial and error. And then it's a lot of trying to get past the doctor's bias as to what is acceptable. Like I remember, to use a very simple example and then I'll be quiet, but most doctors that we've encountered think that he takes an unreasonably large dose of potassium. And then I remember talking to Jake when we were in Ireland at the last conference, and Jake, through his own self-advocacy and self-management, has figured out that prior to going for a workout, you can have a ridiculously large dose of potassium, which most doctors that you run into who are unfamiliar with the disease would say, good God, never do that. You're going to have a heart attack or some other problem's going to happen. And so, again, it's one of the reasons why we're here, is to share those stories of what can you get to in terms of acceptable levels of both dosage and trial and error. And but that article is a great one to start with a lot of doctors that just haven't crossed the line yet. And again, at the end of the day, I personally think that the right diagnosis is unbelievable, but I also think the practical aspect of how you deal with it every day is more important from my simplified judgment, because at the end of the day, we're all trying to live excellent lives. And so the practical aspects of managing the thing are a pretty big deal. So thank you for writing that article.
Yeah, I would also shout out on that article. I keep a copy of it on my laptop.
Thank you so much. No, I'm just kidding.
Right. But there was actually a follow-up on that one question. And one other thing, that did actually make me think that, you know, you might try negotiating with your doc about what would be, for example, the potassium dose that they would be comfortable with. Check labs afterwards. They don't even wait, not even waiting for an event, but just take a dose if they're like, boy, I just, 5 milliequivalents, that's going to be pushing it. Okay. Well, let's give 5 milliequivalents and then let's check potassium. And lo and behold, it's not going to be 7.3. And that may then create some comfort level for your doc to say, boy, you know what, actually, I guess we really could go up because, and then obviously it means you get stuck a bunch, but it does create a little bit of comfort for your doctor. Because your doctor wants to do the right thing. You had a follow-up on that?
I guess I was just thinking in terms of longevity due to our daughter being 8 as well as other pediatric populations, but from a potassium versus, I know the neurologist also brought up acetazolamide, and I guess I was just thinking from a standpoint, thinking back to her concern with renal function, as well as other organ damage. I mean, as we approach it with our neurologists, is there concern or a better initiation point? Like, is potassium better to start with rather than looking at?
So, and this is just my gestalt, and other people would have a probably better feel, but I think that potassium is fine to start with. I think that if your plan though begins and ends with potassium, I think that you're potentially missing a lot of other opportunities. Again, the kidneys accommodate. The body's very good about putting the potassium back where it thinks it's supposed to be. And so it's really hard to capture enough potassium over a long period of time, day in, day out, to be able to really prevent issues. So I think that it's, most doctors are going to be more comfortable starting with potassium than they would starting with, because when they were in residency in the NICU, they were given, itsy bitsy little babies, preemies potassium. And so they're going to be comfortable with doing that more so than the Keveyis or acetazolamide or spironolactone or something like that. Even though I really think that at some point your plan, unless low dose potassium just on a PRM basis, if that controls everything, great, no reason to do anything else. But you certainly wouldn't want to limit your therapeutic options, in my opinion, just because she's 8.
I mean, the short of it is that a lot of us in this room have been taking acetazolamide since we were teenagers, and we're now 40, 50, 60 years old. It's not messing up your kidneys if you stay hydrated. And if you take decent doses of potassium, it's going to make your paralysis go away and not hurt your kidneys. So I think, and certainly in the short term, you want to know if these drugs work. So you know you have the tools, and then you can decide to continue them or not. But you want to have eyes wide open, versus just being paralyzed and tired all the time. So, you have to weigh those risks and benefits.
I would also say that Jake is not the poster child for carbonic anhydrase inhibitors. So don't let how he turned out scare you from your children being treated. So.
Hi, my name is Nikki, and I am clinically diagnosed with hypokalemia, as well as my 16-year-old daughter. I just want anybody that wants to talk to me about their kids, their teenagers, their experience, compare notes, find me. There you go.
Outstanding. And I actually have some questions about my teenage daughter, so I'll be chatting with you later.
So this guy right here, stand up, started on Diamox when he was 6, and at that point they were a little concerned about whether or not it would affect growth, and he's 6. Apparently not. So anyway, if any doctors are concerned about that, you can let them know it doesn't.
He's a more positive poster child than Jake is.
Surely.
Oh, yes. Sorry.
Hi, my name is Vivian, and my son is diagnosed hypo with the calcium channel, and he's also hypermobile Ehlers-Danlos syndrome. And online, I noticed on Facebook, a lot of EDSers also have hypo. Is there any sort of link that you found for that?
Yes. Any other question? No. So we don't understand, or at least I don't understand. Smarter people may very well, but we don't think that the collagen laxity that we see in EDS hypermobile type has really anything directly to do with the ion channel issues that we see with PP. But it keeps coming up, and just, as you work with patients, you just find that lots and lots and lots of patients who have issues with periodic paralysis also have joint hypermobility. And EDS hypermobile type, certainly the most common, and even though we say that, it's 1 in 5,000, the reality is that there's so much overlap with something called benign joint hypermobility syndrome that you're really probably looking at maybe 0.1% of the population or so could actually have that or meet the clinical criteria for EDS hypermobile type. So there's definitely some connection. I'm not exactly sure what it is. Again, we just really don't know. But there is I'm confident there is some connection, and it's because it's just you see it over and over and over again, and there's got to be some explanation for that. Anybody else want to take a stab at the whole EDS thing? Because I'm lost. But again, it was actually one of my patients. I'd never even heard of that. And then one of the first PP patients I was seeing, her husband actually disconnected her lower leg and sort of turned it like 90 degrees and put it back. And they were trying to convince me that her knee was unstable. And I'm like, whoa, you could have just said, I think my knee's unstable. Put that back where it's supposed to be. Don't do that again. So again, you just see it over and over again. Someone had their hand? Yes.
One thing that I've noticed, or that I've noticed a lot reading online, is that people say that, as you get older, you get fewer and fewer instances of the paralysis. Well, in my situation, I wasn't diagnosed until like a year ago, but I've been having symptoms like pretty much my whole life. I can remember things back from when I was like 5. But the weird thing that happened with me is that it hit me really, really hard around 14 or 15 years old, and kind of followed me up to about the point I was 30. And then I didn't have a lot of problems between 30 and 40, and now it's like hit me hard again. And I don't know if that is something that's very common, where people will go a long stretch of time and not have a lot of issues. And then suddenly they have a lot of issues again, or, because everything I've read seems to, like, I should be starting to get better by now.
I am exactly the same way. So you and I need to talk, okay?
Any of my adult colleagues want to take a stab at that one?
So there are recognized connections between hormonal status and susceptibility to periodic paralysis, particularly hypo-PP, particularly the calcium channel mutations, particularly R528H, if you're in that bucket. So one of the best studies on this, very large family in the Netherlands, over 40 affected individuals, huge 5-generation family that Tara Links and her colleagues described. And so many of the women did not experience episodic attacks classical for periodic paralysis, but then in their 40s and 50s began to get permanent muscle weakness, proximal weakness, difficulty climbing stairs, things like that. Whereas the men were much more likely to have the episodic attacks and then develop, unfortunately, the progressive weakness as well. In fact, that one family is one of the strongest conundrums of the fact that it's hard to connect the dots between frequency and severity of episodic attacks and subsequent risk for late permanent weakness. It's because, that seems to break that rule. But of course, there could be lots of subclinical episodes that, don't rise to the level of appreciation that's wear and tear on the muscle. The other hormonal thing is that, and Todd, I'll be curious in your experience, the textbooks say, and a lot of the textbooks are wrong, that more typically, hypo-PP has its onset at puberty and later, whereas hyper-PP presents more commonly earlier. There are exceptions to all of this. There's some nice large studies now with histograms, and it's kind of all over the map. But that was the initial impression, and so there was another hormonal connection there. And I'll actually show you tomorrow our mice with R528H. The female mice are not as severely impacted as the male mice. So there are definitely hormonal things there. We just don't completely understand.
What I think might come out of this is, we talk about papers that say, redefining periodic paralysis, and maybe it's time for a kind of seminal redefining periodic paralysis to break all of these preconceived notions that block diagnosis for so many folks. I think we have to call it. Thank you, Dr. Bell, for your moderation. And I will cede the mic to Linda for our last thing of the, I don't know. Oh, Jeff has where's Jeff? I'm going to cede the mic to Jeff. Oh, does that mic not work?
What do you need?
No, you have to introduce Canine Companions.
I don't know anything
Oh,
about
you don't
canine
have it? You don't know
companions.
yet?
I'm just Steve.
Okay. he's.
Okay, this afternoon, can't believe my computer just died. This is too funny.
I bet it didn't die. I bet you just have to tap it.
Here we go. Unbelievable. Founded in 1975, Canine Companions for Independence is a nonprofit organization that enhances the lives of people with disabilities by providing highly trained assistance dogs and ongoing support to ensure quality partnerships. The assistance dogs that are bred and raised and trained aren't just the ears, hands, and legs of their human partners. They're also goodwill ambassadors and often their best friends. They open up new opportunities and new possibilities and spread incredible joy. We unite people with dogs in a powerful program that leads to greater independence and confidence. Canine Companions, and I might be stealing some of your thunder here, but it's on the notes and I get shot if I don't do them. Okay? Not really, Jake. Canine Companions provides assistance dogs free of charge for adults with disabilities and are partnered with service or hearing dogs to increase independence. Assistance dogs perform physical tasks for children with a range of disabilities to increase independence with support from an adult. Wounded veterans and veterans with disabilities are matched with assistance dogs for greater independence. Assistance dogs are expertly trained and partnered with a working professional in a healthcare, visitation, criminal justice, or educational setting. Today, we are joined by Melinda Sowers, Program Manager of the Southeast Region, and Adele Moses, the Head Trainer for the Southeast Region. Welcome.
Hi, everybody. Hi. So thank you for letting us be the end to your Friday today. We are really excited to be here. Thanks to Linda for inviting us to be here. So as Jeff said, we are with Canine Companions for Independence. We have been placing assistance dogs for more than 40 years. We are a national organization, and we're all throughout the country. So we have 6 regions all throughout the country, and each region serves a certain number of states. We do provide dogs free of charge, and we are an organization that is accredited by Assistance Dogs International. And what that means is that there's a certain standard that we have to live by as an organization that has that accreditation. And that standard really has to do with dogs and has to do with people and how we work with both. So what we're going to do today is just a little bit is, I'm not going to talk a lot because I think probably what you want to hear about is the dogs more than this part. So I'm going to make this part brief. And then we're going to, Adele, who is our training manager, she is going to show you a little bit about what our dogs can do. And then we would love to just answer any questions. We know there are sometimes a lot of questions about just assistance dogs and what they can do, or our organization in particular, so we are happy to take your questions at the end. So just a little bit more about us. As Jeff said, we do have a couple different types of dogs. Our dogs do things like pick up items, open and close doors, drawers, refrigerators. We also do place hearing dogs as well. And really, our dogs are placed to really help someone mostly with physical physical tasks. And so that's what's important to us. That's what we train our dogs mostly to do. And Adele's going to talk a little bit more about our breeding program, but we do breed them specifically for a temperament that's great with working in public. So this is Hobbs, and you can see, we see other assistance dogs out there, and so they certainly have to be a certain temperament to be able to do this work. So definitely breed for that, and we will show you a little bit of that in a little while. So what I want to start off with is we have a quick video. It's animated and much more interesting than me describing our application process and kind of the journey that a person and a dog goes through our organization. So I'm going to ask for the video to be queued up, and then we'll talk a little bit about this after the video is over.
Canine Companions for Independence is a nonprofit that Canine Companions for Independence is a nonprofit that provides highly trained assistance dogs free of charge to people with disabilities. At their national headquarters in Santa Rosa, California, the work starts before puppies are even born. They work hard to meticulously select and pair dogs for breeding. Litters are then raised in homes with volunteers. At the same time, a request for an application is submitted online. As the applicant's paperwork is being filled out and sent, the quickly growing 8-week-old pups are placed with volunteer puppy raisers where they will be cared for and learn basic socialization and obedience skills. Before applicants are accepted to receive a dog, they go through a multi-step interview process, including a phone interview, medical paperwork, and an in-person interview. Accepted applicants are put on a wait list until they are invited to a team training class to be matched with an assistance dog. When the dogs are fully grown, they leave their puppy raisers and return to Canine Companions to work with skilled professional trainers to learn advanced commands and tasks. Then comes the day when the person and dog meet for the first time. Here at Team Training, the newly matched team learn to work together, and training continues until they graduate as new partners. Canine Companions will continue to follow up with the pair for the lifetime of the placement, serving as a support system for the team. Canine Companions for Independence has been providing assistance dogs to people with disabilities free of charge since 1975.
So like I said, I think that can put it really well, instead of us talking a little bit about that. I think Hobbs really liked the video. He seemed to be like super interested up here watching everything that was going on. So with our organization, as it mentions in here, we have a 4-step application process that our folks go through if they're interested in getting a dog from us. Throughout that process, we're trying to figure out what's their lifestyle like, what do they need, what kind of dog would work best for them. Are they on the go a lot? So are they out and about a lot in the community? Or are they someone who stays at home a lot? And that determines what kind of the best dog is for that particular person. And so through that application process, we're really getting a feel for what that person is like. And then Adele will talk a little bit about the training piece, and through the training part, we really are getting an idea about what the dog is like. And so when somebody comes through our process, they're accepted, and they go into that team training class, that's when we really start to look at, okay, which dogs do we have and which people do we have, and really start to make that really perfect match between that person and the dog. And we're really looking for a dog that will work for them, that will increase their independence, that can really help them with the different physical tasks that they need. So just to illustrate that, it's easy for me to kind of say it helps with independence, but I've got just a couple of stories that just kind of illustrates a little bit more about what our dogs can do. So we had a family that came through our organization years ago, and they were raising children, and mom had a disability, and dad worked, and mom wanted to be as independent as possible. She wanted to raise her boys and be independent, and she came and got a dog from us, and the dog did multiple things for her at home. The dog helped her with laundry. So the dog would actually, like once it cooled off, get items out of the dryer for her, put the items in a laundry basket, and then the dog would actually pull that laundry basket from each room in the house for her, and she would then be able to get the clothes out, fold them, put them in everybody's rooms. The dog was able to help with different things in the kitchen, so opening the refrigerator for her, helping her with getting dinner ready. And years later, she told us what an impact the dog had had on her and her boys, and her whole family, as she, really raised her kids, was able to be independent and do things around the home. And then we have some that we place some hearing dogs, and the dogs can alert them to different things. So there are a lot of different ways in which our dogs can help. And so Adele's going to talk a little bit now about what those ways are and show you what our dogs can do, talk a little bit about the dog's journey. We do have some brochures in the back and bookmarks, and so with this whole application process, if anybody is interested or has questions at the end, we certainly will stick around, but want to give you a chance now to see a little bit about what the dogs can do.
All right, so I have Hobbs with me, and he is a dog that's currently in professional training. He has completed 6 months with us in the professional training. Prior to that, he was with a puppy raiser for the first 18 months of his life. So our puppy raisers do a lot of socialization with the dogs. They take them out to different places, get them used to all sorts of different environments, get them riding in cars, listening to different sounds in the environment, getting around all sorts of different people, adults, children, cats, other animals. And so by the time they turn in for their professional training, they're pretty used to everything that they could experience in life. Then when they come into professional training, our trainers teach them some advanced commands that really help our graduates in their lives. So the biggest command that is requested of us is for the dog to retrieve dropped items. So I have a couple things with me here. I have a set of keys. Hobbs, release. Come on. Get. So he will pick those up for me, maybe. Come on, bud. Don't get. Good boy. Sit. And it's important for our dogs not just to be able to pick him up, but to be able to hold on to the item until I'm able to take the item from him. It does no good for me if he picks up the items and as I'm putting my hand down, he spits it out. So we do train our dogs to hold on to the item until the person can get a good grip on the retrieve item, and then tell him to, Hobbs, give. Good boy, thank you. They can pick up a variety of items, anything as small as a set of keys or a credit card, to things that are about 5 pounds. So he can pick up my keys, he can pick up a cell phone, he can pick up, let's see, what do you pick up, buddy? Clothes off the ground, help with laundry, those kinds of things. He can pick up a water bottle for me. Another task that our dogs are trained to do is to push with their nose. So they can either push drawers closed or cabinets, but they can also hit the automatic buttons that open doors. So he can do that. He also learns how to tug things open. So he can tug a door open by grabbing a strap with his mouth, and then he'll back up with it in order to hold a door open. So I wouldn't have to hold the door and use my strength for that. I can have him do it and help me through. Let's see, Hobbs, heel. Good boy. So he's about 2 years old, and he is ready to go into our next class of team training. You saw a little bit about that in the video, that we have 9 people coming on Monday to start working with the dogs and get matched with one of these great animals. He and 13 other dogs are going into class. We always put more dogs into a class than we have people coming, because we want to make the best matches, not just the matches that we can make. So he'll be working with a variety of different people. We'll see how he interacts with them, how he responds to them, what they like, what they're looking for in a dog, and we'll be able to match him with someone in order to increase their independence. Oh, yes, I was going to talk about the breeding a little bit. So we breed our own dogs. Kind of the hallmark of our program is that we know everything about these dogs from birth through their entire lives. We have over 30 years of breeding program in order to breed for the best temperament and health that we can in our dogs. So we do all sorts of checks on them to make sure they're healthy and appropriate for a working role. We look at their hips, we look at their elbows, their hearts, and their eyes. And any dogs that have a problem that would shorten their working life are removed from the program. Those dogs end up going to wonderful homes. The puppy raiser of the dog has the first opportunity to adopt the dog, and then otherwise we have a very long list of people who would like a dog from us. About a little over 50% of our dogs actually make it into a graduate role. Because of our very high standards, we want to make sure that the dogs are healthy, happy working, that they're really enjoying this role. We wouldn't want a dog to be uncomfortable in public environments or afraid of things that they might be encountering on a daily basis.
Yeah, so a couple of other things that our dogs can do. So of course, I think, you know, the physical tasks are great, and so some of the other physical tasks are they can pull a manual wheelchair, they can turn light switches on and off, they can do what we call counter transactions. So sometimes if it's difficult to access a counter, like at a grocery store, they tend to put like all the stuff in front of counters. And so sometimes it's difficult to access that. The dog can actually put their front two paws up on there, give a credit card, take a bag, carry a small bag, like Adele said, under like 5 pounds. Really a lot of different things in public. Push elevator buttons, push plates to open accessible door buttons. All of those things just to make it easier when you are moving around. They can work with equipment, so they can work with power chairs, they can work with manual chairs, they can also work with somebody who is walking. So we do train them in different ways so that they are set to work with somebody no matter what kind of equipment they're using if they are. And that's something that we find out during the application process as well. Those physical skills are really important to our graduates. However, I would think anybody who has an assistance dog in this room, I know Linda would probably attest to this as well, is that the companionship can be really great too. And obviously having that bond with an assistance dog, really having a dog there can really make a difference as well, just on a day-to-day basis. And it's just a super bonus that they can do a lot of great physical tasks for someone. One of the other things that our dogs can do, it's a newer command that we're teaching now, is called cover. And so for someone that may need some sensory input, the dog is able to put their front two paws over the person's lap and kind of lay across them. And that has been a newer command that's been really helpful for our folks for various reasons. Some folks that have different disabilities that need sensory input, need that calming effect. That's been a really good one. So we do serve kids ages 5 and up. For anyone that is under 18, we usually have a parent or someone else who is involved in working with the dog with them. And then there's, that team where someone 18 years of age or older is handling the dog on their own. So we can accommodate 5 and up, and it just depends on, what the person needs in terms of what they're looking for. Yes, question? Oh, you want to, let's see, do we have a mic?
My name is Niva Jareth. I'm starting a neuromuscular clinic here in Orlando, so for Advent Health, and it's a very popular clinic. It's, I think, my 2nd month, and so I have cards, and I've sent some in the back. So for those patients that don't want to travel far, I've had extensive background, and you guys can come see me. But I think this is a great idea. And one of the questions I have for you, for my patients, is that do the physicians need to help with the application process? That's one question. And the second question is, do you give preference to patients who are in wheelchairs or more significantly disabled than others?
That's both great questions. So I'll take the first one. So with our application process, as we noted in the video, there's a 4-step process. Step 3 of that is actually reference forms. So yes, it's really helpful when physicians or therapists who are working with our applicants are able to give us good information about what they need. We have specific questions that we ask them about what their needs are, what their disability looks like, how they would handle our team training class that we've referenced. And so all of those are great questions, and that really just helps us get an outside opinion. So yes, that would be great. And a lot of times folks will take that to their doctor, ask them to fill it out, and then they'll send it back to us to provide some much needed information, just another part of the application process to really help us understand who this person is and what they need. The last step in that is a personal interview. So we do see folks before we place them. They come to our center in Orlando. We're from the one in Orlando, but again, there's ones all over the country depending on where you are. And you will actually meet with us. You'll see the center where we work. You'll actually get to handle a dog. So you'll actually get to take one of these dogs and handle them so you get an idea of how it is, as well as we're able to see what your handling is like. Again, just another piece of that puzzle that helps us pick the right dog. So that's really, really important. In terms of our waiting list and preferences on our waiting list, we really do a first come, first serve on our waiting list. Now, what happens is if we have if we have 5 people at the top of the list, but we only have dogs for 3 of those, of course, some of those folks that have waited longer may not get a dog at a specific time because we have to have the right dog for people on the list in order to place them. So we try, so if there's that person that's at the top of the list, they've been waiting the longest, we select them and put them into class, and the person underneath them, we decide, you know what, we just don't have the right dog right now, so they would actually be considered for the next class. So we try to do first come, first serve, but again, that's really dependent on whether or not we have the right dog.
How long is the waiting list?
That's a great question. So the waiting list can be anywhere from about 1 to 2 years after the application process. It can vary a lot based on a lot of different factors, and some of that is what type of dog you need, what specific things are you looking for. Also, your availability. Sometimes people will say to us, I am not able, we have 4 classes a year at our centers, so if they say, I can only come in August, I can't come any other time for the year, that can increase somebody's wait time. So the wait time varies a lot per person, but on average about 1 to 2 years.
To add a little bit about the diversity of clients, we have a wide variety of dogs that we train. So some dogs are very slow movers, they're very deliberate with their skills. Other dogs are very fast and quick to do their commands. And so different people are going to need different types of dogs. Some might need that slow-moving dog that waits until they hear the next command. Others might want that fast dog that can keep up with them. So between wheelchairs, power chairs, individuals who are ambulatory, we're able to serve a wide variety of placement types, and there's not really any sort of preference one way or another.
So as we mentioned, there's no fee for our dogs. Our graduates are responsible for all the care of the dog after the placement. So all the vet care, the food, all of that after the placement, but no fee to get a dog from our organization, no fee to come to our class. All of our regions, except for our region in Ohio, have dorms on their campus. So we do have graduates stay on our campus free of charge for that 2-week team training class. Our Ohio region is getting ready to open a new campus, and so hopefully soon that one will also have dorms. So we do provide that when folks are staying on campus for those 2 weeks to make it easier for folks to have a place to stay, be right there on campus, be able to train for 2 weeks with the dog, and be able to let us help them care for the dog and get acclimated before they actually take the dog home with them. Our dogs are all able to go out into public and have public access. Under the ADA, as many of you probably know, having a dog go into public is a possibility if the dog is doing a physical task for someone to help with a disability. So push plates for doors, picking up items, pulling a wheelchair. So all of our dogs have gone on multiple field trips, been evaluated, done travel in cars and different, a bus. So we've done all we can when we have a dog at the center to really evaluate how they would do in a public setting and make sure that they would be completely appropriate on a stage, on a bus somewhere, traveling with someone, going to work with someone, going to the grocery store with someone, all of those things to make sure that they would be appropriate in that setting.
It's getting near 5 o'clock. What I was hoping maybe if someone with a service animal for periodic paralysis wants to just mention how it helps them, because I think that when we started this conference, nobody had a service dog, and there were 70 people, and now there's 200 people, and I see 6, 7, 8. So it's getting to be a thing, and I think that if someone has experience. So yeah, okay.
I just got mine. He's still in training, but it was a big decision. It took me a couple of years to decide if I wanted to have a companion with me all the time. It finally got to the point where one day I was by myself, started to become symptomatic, and I realized that the dog could save my life. And so I decided to take the big step, and it's, even though he's not fully trained, it's been reassuring because he's comforting, if anything. So hopefully, the next episode I have, we will know if he has, his training is taken, but, and I'm kind of the guinea pig for my family. If this goes well, then the other people in my family that have periodic paralysis is also going to get a dog.
Great. Thank you.
Yeah, I think that's so true, and I think you make such a good point. It is a very big commitment. Just thinking of having the dog with you consistently and all of the care, there's so many benefits, but it is definitely a decision, that folks really think about for a while before doing that. So it's great that you did. So good luck. Sounds like it's going well.
Yeah.
Monica's dog is being trained to tell her before she has an episode. Can you do that with your dogs, or is it just to be able to help with tasks?
Great question. So we only train for the physical task at our organization. However, I know I've talked to Linda a little bit about this as well, and I think that we certainly, if someone had the need for the physical task, but also was interested in after that training a dog to do, to alert, that would be something that we certainly would not be opposed to having dogs do afterwards. It's just not something that they would leave our center doing. It's not something we do, but it is something that if someone were to get a dog from us, need the help with some physical tasks as well, it certainly could be something that could be done after.
My name's Mike Zagrabic, and my wife has PP, and we just started with the training dog, and it's something that as a caregiver has been a big help. It's taken a lot of the pressure and the load off and has helped me be more attentive to my wife's needs with the dog helping. So not only is it instrumental to the person who has the dog, it's instrumental to the caregiver as well. Don't be afraid to, as a caregiver, you need to be an advocate for yourself as well. So don't be afraid to ask your partner to take them steps forward to this wonderful program with the Canine Companions. It has been a great help for me as a caregiver, and I highly suggest it to you as caregivers as well to take that step forward with your partner.
Yeah, thank you. That's a super good point, and I think that it does help. I think, not only does it change the life of the person that gets the dog, but so many times do we see how much it changes, relationships and families and just, everything in terms of the family system. So you're right, absolutely. How about other questions?
Is there someone back there? Yes? No? Someone?
I guess mine's not really so much of a question, but a comment, since we've had service dogs for several years now for this condition. When my son and I both are hypos. When my son was first symptomatic at age 9, It was very hard to control. He would wake up in the mornings and try to get out of bed and face plant because he didn't realize he couldn't walk. And then in a very short amount of time, he'd get clear to the point where he'd be in full body paralysis and couldn't speak. So he would literally lay there sometimes an hour or more before I would realize that, hey, he's not getting up, I need to go check on him. So then we would have a lot of trouble getting him to recover quickly. So that's when we first started looking into a service dog, and we got one that would alert to falls, and she slept with him. So every time in the morning when he would get up and face plant, she would bark and let me know that I had to get back there right away. And it was so liberating for me because I didn't have to constantly, every minute, be going back and listening by the door, peeking in and looking at him. I could go out and do things in the yard, leave the window open and hear her bark and know to go get him and get his fizzy in him.
Now they have Google Watch, right?
I haven't tried the Google Watch, but...
My dad has it. He took a spill at 911. Yeah, it was pretty good. I think it's 5 o'clock. They have to get the room ready for dinner. So when is tomorrow start? 8:30 a.m. So yeah, let's let you know if you're going to dinner with Strongbridge, it's tonight, and otherwise enjoy Orlando, and just dinner's in here, right? Yeah. So freshen up, whatever you need to do. Is it 6? When is dinner? 6:30. 6:30? 6 o'clock. So in an hour from now, be back here for food. All right. Thanks, everybody. Thanks, Canine Companions.
Thank you.
Thank you.
Thank you, folks.